Hello, everyone. Welcome back to The Issue on the ASGCT Podcast Network. I'm your host, Emily Walsh Martin, and today's guest is my former colleague, Sonal Gupta. Sonal is the Senior Vice President, Head of Clinical Development at Affyimmune Therapeutics, and we use our time together to continue to explore the challenges of cell therapy clinical trials and how to move patients thoughtfully and quickly through the steps required to receive therapy in aggressive solid tumor cancers. But first, we've moved straight from pumpkin spice season into ASGCT annual meeting registration season. So hop online and join ASGCT, then register and take advantage of great discounts, which are available now. Then we'll see you May 13th through 17th in New Orleans. Can't wait to see you. Hello, and welcome to The Issue on the ASGCT Podcast Network. Sonal Gupta, Senior Vice President and Head of Clinical Development at Affimmune, which is a cell therapy company that's focused on CAR T development for solid tumors. I'm so happy to have you here today. We've got so much to talk about, but before we dive in, I know you very well from our time where we overlapped at CRISPR Therapeutics and as well from our time at Affimmune together. But could you share a little bit about your background and current role? Because I think your background and path is going to be very interesting to our audience.
Thank you, Emily, and it's my pleasure to actually be here. Thank you so much for including me. And again, I've always enjoyed working with you and talking to you, so this is great. So as you mentioned, I'm the Head of Clinical Development at Affyimmune Therapeutics. I'm a physician from India. I have a PhD in cancer biology and immunology, and I work now in the Boston, Cambridge area since 2006, originally as a postdoctoral research fellow, and then through a series of companies which have become smaller and smaller. And right now I work for a small company called Affyimmune Therapeutics, as you mentioned. You and I worked together at CRISPR Therapeutics. I was part of the early development team there working on Casgevy. And I've worked in rare diseases and cell and gene therapy now for close to a decade. So it's my pleasure to speak to you.
Yes, absolutely. Yeah, it's I think what's amazing about this whole industry of cell and gene therapy is the fact that people that you work with once you often get to work with again, which I think has all sorts of benefits for how we move forward on these projects, on complicated projects with good teamwork. But let's dive in. I would love to hear a little bit about how Affyimmune is approaching solid tumors. CAR T has been established in blood cancers now for a while, but it feels like the progress in solid tumors is just starting to emerge. So can you talk about the Affyimmune approach?
Of course, yeah, happy to. So we are not the only cell therapy or CAR T cell companies trying to treat solid tumors. Like many other CAR T cell companies, we have a platform. Our platform allows us to very coolly track our CAR T cells. So we use an FDA-approved technology called gallium dotatate PET-CT to be able to track our CAR T cells in patients. It's basically biodistribution, but in people, right? Usually most people do it in animals. We can actually do it in people, which is very cool. It also helps avoid repeated biopsies on these patients. Secondly, if you know that a lot of toxicities that you see in solid tumors, as well as hematological cancers, are because of high affinity CAR T cells. And so what Affyimmune, which is where the name Affyimmune comes from, we affinity tune our CAR T cells to the target. In this case, it's ICAM1. And because we affinity tune, we optimize the binding of our CAR to the target, ICAM1, which helps us avoid serious toxicities that you see with other CAR T cells. And so that's the basic platform of Affyimmune. Our first indication is thyroid cancer. And the reason why we selected thyroid cancer, especially the more advanced thyroid cancers, is because there's a high unmet need. It's not a very well-studied cancer. And secondly, thyroid cancers express almost 100% ICAM1. So every cancer patient we have tested for ICAM1 actually expresses ICAM1. So it allows us to not require a companion diagnostic.
Right. And we'll come back to that, I think, when we start talking about, the entire sort of process of going from, identifying a patient to actually giving them the therapy, and how there's so many steps in this process. So every step that you can strategically find ways to be smart about benefits the patient at the end of the day, right? So it goes without saying, solid tumors are often, very aggressive tumors, and they progress rapidly in patients, particularly some of the ones that Affyimmune's working on. So sometimes by the time you have a diagnosis and you've screened a patient to include them in a trial, you're sort of racing against the clock, the cancer's progression clock, to do the quality manufacturing, appropriate release testing, lymphodepletion, and administration so that the therapy has the best chance of success. And what I thought you and I could talk about is sort of take our listeners through all the different perspectives of how this race occurs, and sort of what the streamlining of the process can look like, so that you move from, vein to vein very quickly, so to speak, and what sort of cross-functional and cross-organizational coordination has to occur between and within the sponsor, right? The site, the CDMO, and regulators. And I thought maybe the best way to start with this is to talk about things from the patient's point of view. So can you sort of describe the typical patient journey in the trials that you've been part of, and sort of like what that patient goes through as they're, realizing that they have a cancer that might be appropriate for a trial that you've worked on, and how those steps sort of occur?
Of course, absolutely. So I'll give you an example of one of the most aggressive cancers that we're trying to treat. It's anaplastic thyroid cancer. Usually, if you Google anaplastic thyroid cancer, it's very scary. The median overall survival is less than 5 months after diagnosis. So it's a race for these patients. There's very few options. So if I was a patient with anaplastic thyroid cancer, I would be very scared, and that's what these patients are. And then so when these patients are first diagnosed in a community setting,
Mm-hmm.
they immediately need to be to refer to one of these major centers, example, MD Anderson, City of Hope, and Northwestern, because that's where you have the oncologists who have the most experience with these cancers.
Right,
And
because these are rare cancers,
the
right?
rare
So
cancers,
their first stop of a patient, in their community hospital, they may be able to say, I think you've got this cancer, but like after that, they sort of have to refer them to a different center.
exactly. Right. And referring them to these major centers or centers of excellence as soon as possible is the most important thing you can do. Because the only way that you can manage anaplastic thyroid cancer currently is multimodal. You cannot just use surgery or radiotherapy and be done with it. It's basically attacking them from every side. And that's what these big centers really do really well. And then when these patients come to these centers of excellence, right, they are usually treated with something. I call it throwing spaghetti at the wall, because there's very little that's actually approved for these patients. So they will
Right.
go through off-label treatments, maybe something. There's only one drug treatment actually approved for these patients. They will, if they have that particular mutation, they will be treated for that mutation. But after that, there's nothing else except for, honestly, our experimental drug.
Wow.
And
Yeah.
so, but because it's a CAR T cell, it means that, and it's an autologous CAR T cell, it means that those patients have to first go through a procedure called apheresis to collect that apheresis product. Then the process of manufacturing, as you mentioned, the process of release, right? And then the actual patient gets lymphodepleted and then gets the CAR T cells. The challenge with anaplastic thyroid cancer is when the cancer starts, you want to get to these patients before the cancer starts doubling every 5 to 7 days.
Mm-hmm.
Because at that point, there's nothing that can actually help these patients.
Sorry, can you say that again? The cancer in this case like doubles every 5 to 7 days?
When it starts really progressing rapidly,
Wow.
it starts doubling every 5 to 7 days. And there's really, at that point, there's nothing that can really help those patients. So you want to get to,
Wow.
we need to get to those patients before that. And so unlike, when we talk about less aggressive cancers where vein to vein becomes really important, for our cancer, it's the way our head of manufacturing puts it really well. It's opportune manufacturing for opportune treatment. So what we're offering especially in our pivotal study, is going to be what we call apheresis at risk or pre-enrollment apheresis. So our goal is that when these patients are first seen by the sites, the apheresis product will be collected, and the patients will go on their first treatment. And when these patients just start progressing, we trigger manufacturing and we dose them, because we really want to get to these patients before they reach that phase which I was talking about, where the cancer starts doubling every 5 to 7 days.
Right. And just maybe double clicking on that, I guess the other benefit of the sort of apheresis, sort of perspective apheresis, is that some of the treatments that these patients might go on might actually impact their white blood cells too. And so if you can apherese and collect the white blood cells early on, those might be, I guess, better in better shape to do what has to be done once they're re-administered post-manufacturing.
Yes. Hypothetically or theoretically, I should mention, although there has been literature out there, you would get higher quality CAR T cells, also less exhausted CAR T cells. So that's the other benefit. So it's getting to the patients at the right time, and then actually making higher quality, less exhausted CAR T cells.
Yeah, no, that makes a lot of sense. And I mean, I think it goes without saying, right? These are patients who are faced with this pretty terrible cancer, pretty fast progressing cancer, and they're also trying to manage their treatment, but as well their life. And so, I think it's also probably something you're having to work around the patient's quality of life as well as you try to help, provide them with an option here, right?
Absolutely. Right. And just think about it, right? Some of these patients are older patients. They usually tend to be in the 60s or 70s. We've had younger patients also. And some of them just want to get to the next milestone,
Yeah.
a child's wedding, a graduation, right? And so being able to offer them this chance of having an extended lifespan or an overall survival increasing, that's the beauty of offering them CAR T cells, because it's a one-time treatment.
Right. So let's maybe take a step down the path, if you will, from the patient to the site and the PI involved in these trials. What are they trying to manage internally? To sort of move the patients along, get them identified, and confirm that they're appropriate for the trial, and then move them through the steps of treatment from apheresis to lymphodepletion and then therapy.
Absolutely. So usually what happens is when these patients tend to be referred to these major centers that we are working with. And then depending on the center, what usually happens is, as I mentioned, it's multimodal. You end up having surgeons, oncologists, endocrinologists, radiologists, interventional radiologists, pathologists all work together to come up with a treatment plan, because the goal is to treat these patients as soon as possible. And so that's the first step that usually happens. Now that our study has now treated actually quite a few patients, it becomes an option for these patients. What we used to do in the past was we used to test the patients for ICAM1 positivity on archival biopsies. Now we know that almost 100% of our patients express ICAM1. We actually took it out. It was adding time, which these patients don't have. So when the patients come in, they're considered fit for the study. They are screened to confirm eligibility. Their apheresis product is collected. It is cryopreserved, and it goes into manufacturing. And once the product is released, the patients actually get lymphodepleted, and then they get the CAR T cell infusion. So our goal is to, from the time the patient becomes eligible for the study to the time the patient actually get infused, the goal is to make that happen as soon as possible. And it really helps that these patients have champion investigators. The oncologists, endocrinologists, they know that these patients have no other options. They are their biggest champions. And I think that really helps a lot. And then the patients themselves and their families also become advocates. And that's very common for rare diseases, and same with rare cancers. So that's the journey that the patient goes through when they're seen at these centers.
Yeah. And so, I think from the sites I guess, perspective, they are managing these patients sort of continuously, right? Because some of the therapies they're on may make them, a little bit ill or cause sort of hiccups, I guess, in the path to sort of move through, say, a lymphodepletion or something like this, where you're having to constantly monitor and figure out, okay, this is a patient who's, progressing on the current therapy, but is not yet in a good enough, I guess, health, if you will, to be able to go through lymphodepletion. How do you coordinate from your perspective those interactions with the investigators to try and manage and help them figure out how to move through the steps?
So we do it in multiple ways. I'm the medical director also, so I'm in regular touch with the investigators. I speak to them weekly sometimes, and especially before and during the patients getting infused with the CAR T cell, it's almost daily. So we
Mm-hmm.
stay in touch. We also have what we call eligibility checks. We do it at screening, we do it prior to lymphodepletion. We also perform that prior to infusion, and that's to ensure that the patients are fit for the study. So the goal is that the patients can tolerate lymphodepletion and CAR T cell infusion. So it's a very close relationship with the investigators. They're like family now, and that actually helps, I think, best manage this for the patients also.
Yeah. And it's interesting, right? Because you mentioned this at the sort of top. AffyImmune's not a huge organization, right? And so in a lot of ways, the rareness of this disorder, the fact that there are a certain number of centers of excellence, allows you to have you as a single medical director to have that sort of constant contact so that you can help move a whole trial forward.
Absolutely. And our investigators also have a lot of interest in clinical research and translation.
Mm,
So it helps that they're not just looking at the patients from the clinical perspective. They're also interested in the correlatives. They're interested in the biomarkers. So we try to do this holistically so that we learn something from every patient, which we then apply
right.
to.
Right. And that's something that's come up on past podcasts where we talk about these things, and actually segs us nicely into sort of the next section of our conversation, which is every patient is different. They will have seen different past therapies. They will have, immune systems that are in differing states of exhaustion or not. And so every single manufacturing is in some ways unique, right? Like, as much as possible, you try to have predictability and consistency in the process, but your starting material is a different patient every time. And so I'm curious, like, how do you see it on the clinical side, the interactions with your head of manufacturing and the CDMOs involved with the process?
So we are best buddies. We have to be because these are autologous CAR T cells. They are, they're very precious. Starting from the apheresis material, you do not want to make the patient go through another leukapheresis, right? So it's how do you the collection itself, the shipments, the logistics, the cryopreservation, but also understanding because we are also working with the clinical team works with the CMC team to characterize the apheresis product, the drug product also, and anything in
Mm-hmm.
process, because we're trying to understand why one patient had a great response and one did not, right? So it becomes, and from their perspective also, they look to the clinical team that what was different about this patient. Was there a reason why, for instance, we had a lower yield of CAR T cells? Was there a reason
Right.
why one patient did really well? There may be two patients who look so similar on paper, but they have different journeys, right?
Right.
And so a lot of that, and people are so complex, and cancer is so complex. So, but what we try to see is trends, and then we also try to in this and in the data set that we do have, we also try to see their correlations. And so that's why it's a give and take between CMC as well as between clinical. They cannot be separated. They have to be very closely intertwined, and it has to be a back and forth communication. And we at Affimmune we do that very well.
Absolutely. Yeah. And I mean, not to put too fine a point on it, you're doing this also, especially in a phase one trial, you're right. You're doing this while you're dose escalating in a lot of cases, right? And so you're trying to see these patterns, and some of them may be, patient to patient variability. Some of them may be, the dose isn't quite there yet. And so it must be incredibly complicated to try and map back and understand your data, at least until you're at, the dose that you want to take into a phase two trial, right?
Absolutely. And I think it's also very important not to base any conclusions on small ends, right? So and
Right.
that's where trends become really important. It's so easy to think, oh, this patient did not respond, probably because of this.
Right.
Because there may, especially if you have another patient who did respond, even though they were similar to the first patient, right? So I think the power in data and data sets becomes really important. And so the more patients you have, which is not easy in a rare cancer, the stronger your data set becomes. And the stronger your hypothesis actually become also.
Right. Right. Well, and it's also, I think, complicated just from the fact that you guys are working in solid tumors, right? And I could be wrong about this, but my sense is that with blood tumors, we're starting to understand sort of, if you will, the rules of the game of what's a good looking apheresis look like. These kinds of things, we're starting to maybe have a hint to what is true there, but solid tumors, the rules may not be the same, right?
And solid tumors are more complicated, right? I think I'm not to try and simplify this, but with hematological cancers, you inject into the organ, right? So it is the blood. In
Right.
solid tumors, there are so many challenges. You first is trafficking, because you're still giving your CAR T cells by IV infusion, making sure that the CAR T cells actually reach the tumor, they infiltrate the tumor, and then they persist in the tumor.
Right.
Those are the big challenges. And then keeping in mind that the tumor microenvironment is heterogeneous, it's complex, and it is very often inhibitory. And it's not easy to study without a biopsy, which is not easy on the patients, right? So that's why what is amazing about Affyimmune's product is we can actually track our CAR T cells in the tumors. And that is amazing because we actually see as I call it, lighting up like a Christmas tree. So we see the tumors light up, especially
Yeah.
in patients. And that gives us confidence that our CAR T cells are actually reaching the organs where they're going to be active.
Right. Yeah, I think that is sort of a game changer if you can have some sense that you at least got to the right spot, right? And how long you're staying in that right spot. So, how do you guys think about the sort of next phase of development, given all these complications? When you're going into a phase two trial, one presumes it'll be more patients. How do you think about that next stage of development?
Absolutely. So I think once you know that your CAR T cells get into the tumor, you know that there is a response there, or at least a stabilization, some shrinkage of a tumor, then the focus should actually shift to what makes the right patient, right? Because
Mm,
your drug is not going to work on every patient, but there's a subset of patients where your drug will work. How do you identify that? And that really is becoming the focus now as we move forward, because we know we have a signal, we have trafficking, we have infiltration, we have shrinkage. How do we do this consistently? Which is what you want to do prior to going into your registrational study. That's where we are. Also, not modifying this paradigm of dosing, which really is based on hematological cancers, and making it fit for our particular cancer, is what we're going to focus on in phase two, which is also going to be hopefully a registrational study.
great. And so tell me a little more about optimizing sort of dosing compared to say a blood cancer approach.
So mutation status, for instance, there are certain
Hmm.
mutations. ICAM1 is obviously has its own regulation mechanism. There's patient internal factors, there are external factors, example, drugs, which could have an impact on ICAM1.
Yeah,
And so what we are doing now is, as we're doing a meta-analysis to try to understand what are those patient factors, both internal and external, which could have an impact on how a patient responds. Because the goal is to use that data to tighten our inclusion-exclusion criteria in the next study.
got
And
it.
that's what we're working on right now.
That makes a lot of sense. And best of luck to you and the team as you try to sift through what I can only imagine is just gobs and gobs of data to try and make sense of it. We're at the end of our time together, so I get to ask my absolute favorite question, which is if you can wave a magic wand and fix one thing for cell and gene therapy development, what would it be?
Oh, that's a great question. Let me think about it. I wish we could just cure cancer. And I know that's a very broad statement because every cancer is different.
Yeah.
But if there's a magic switch, and if there's a magic switch, right? That's what earlier CAR T cells did for hematological cancers. We need to find that key for solid tumors. It may be it may be 2 doses of CAR T cells. It may be 3 doses of CAR T cells. We don't know what's going to work for solid tumors. There's early data out there that more than 1 dose of CAR T cells actually needed for solid tumors. And that's something we're also exploring. And just finding that dosing paradigm and when's the right time to actually treat those patients so that we're getting in early. If I had a magic wand, that's what I would have an answer to that.
Yes. No, I like that. Yes. And I think in the near term, if I had a magic wand that I could give to you, I would love a way to visualize all the data that you must have collected by now to
Right.
help you sift through and figure out what might be the clue to get to your magic wand.
So I
So
appreciate it.
thank you so much, Sonal, for coming on the podcast. It's been wonderful talking to you, and really, really appreciate your insights. And best of luck to you and the team, and to the patients that you're hoping to help.
And thank you so much for having me on your podcast. I really had a great time.